Hi everyone, welcome back to अनदर वीडियो। तो आज सिर्फ हम रीडिंग करेंगे एंड रीडिंग करेंगे लेनिंजर प्रिंसिपल ऑफ बायोकेमिस्ट्री सिक्स्थ एडिशन। ओके, तो इसमें हमारे लिए फर्स्ट चैप्टर जो होगा द फाउंडेशन ऑफ बायोकेमिस्ट्री। ठीक है, हमें पहुंचना है कहाँ तक भाई? फार्माकोलॉजी तक हमें पहुंचना है। तो उससे पहले हम ऑर्गेनिक केमिस्ट्री में मास्टरी चाहिए, उसके बाद बायोकेमिस्ट्री, उसके बाद आपका एनाटॉमी, देन फिजियोलॉजी, देन आपका माइक्रोबायोलॉजी, देन आपका पैथोलॉजी, देन आपका फार्माकोलॉजी आपका आएगा। दिज आर द सीक्वेंस। ओके, ये कुल मिला करके सभी बनेंगे, तभी जा करके फार्माकोलॉजी का जो मसाला होगा वो तैयार होगा। ठीक है, तो इसमें बायोकेमिस्ट्री का हम डिफरेंट एंट्री लेवल बुक से ही पढ़ेंगे एंड इससे भी पढ़ेंगे। ठीक है, कुछ कुछ चीज़ें हैं जो बेसिक बुक में होते हैं एंड कुछ कुछ चीज़ हैं जो एडवांस बुक में होती हैं। उनमें से लेनिंजर एक एडवांस कैटेगरी का बुक है। तो देखते हैं द फाउंडेशन ऑफ बायोकेमिस्ट्री। ये हो गया हमारे लिए चैप्टर नंबर 1। इसमें हम सेलुलर फाउंडेशन देखेंगे, केमिकल फाउंडेशन देखेंगे, फिजिकल फाउंडेशन, जेनेटिक फाउंडेशन, इवोल्यूशनरी फाउंडेशन। तो कोशिश करेंगे 2-3 पेज पढ़ने के लिए। ओके, सो इसमें पूरा रीडिंग सिर्फ होगा। ठीक है, अबाउट 14th billion years ago the universe come arose as a cataclysmic explosion of hot, energy-rich subatomic particles. Within seconds, the simplest element जो कि हाइड्रोजन एंड हीलियम होता है वे आर फॉर्म्ड। As the universe expanded and cooled, material condensed under the influence of gravity to form stars. Some stars became enormous and then exploded as supernova, releasing the energy needed to fuse simpler atomic nuclei into the more complex element. Atom and molecules formed swirling masses of dust particles, and their accumulation led eventually to the formation of rocks, planetoids, and planets. Thus, were produced over billions of years. Earth itself and the chemical element found on Earth today, about 4 billion years ago, life arose. Simple microorganism with the ability to extract energy from the chemical compound and later from sunlight, which they use to make a vast array of more complex biomolecules from the simple elements and compounds on Earth's surface. We and all other living organisms are made of stardust. Biochemistry asks how the remarkable properties of a living organism arise from the thousands of different biomolecules. When these molecules are isolated and examined individually, they conform to all the physical and chemical laws that describe the behavior of a inanimate matter, as do all the processes occurring in living organism. The study of biochemistry shows how the collection of inanimate molecules that constitute living organism interact to maintain and perpetuate life, and animated solely by the physical and chemical laws that governs the non-living universe. Yet organism possesses extraordinary attributes. properties that distinguish them from other collection of matter. What are the distinguishing feature of a living organism? A high degree of chemical complexity and a microscopic organization. Thousands of different molecules make up a cell's intricate internal structure. These include very long polymer, each with its characteristic sequence of subunits, its unique three-dimensional structure, and its highly specific selection of a binding pattern in the cell. System for extracting, transforming, and using energy from the environment, enabling organisms to build and maintain their intricate structure and to do mechanical, chemical, osmotic, and electrical work. This counteracts the tendency of all matter to decay towards a more disordered state to come to equilibrium with its surrounding. Defined function for each of an organism component and regulated interaction among them. This is true not only of a macroscopic structure such as leaves and stem or heart and lungs, but also microscopic intracellular structure and the individual chemical compounds. The interplay among the chemical component of a living organism is dynamic. Changes in one component cause coordinating or compensating changes in another with the whole whole ensemble displaying a character beyond that of its individual part. The collection of molecules carries out a program, the end result of which is a reproduction of the program and self perpetuation of that collection of molecule in short आपका life. Mechanism for sensing and responding to alteration in their surrounding. Organisms constantly adjust to these changes by adapting their internal chemistry or their location in the environment. A capacity for precise self-replication and self-assembly. Figure 1C. A single bacterial cell placed in a sterile nutrient medium can give rise to a billion identical daughter cell in 24 hours. Each cell contains thousands of different molecules, some extremely complex, yet each bacterium is a faithful copy of the original, its construction directed entirely from information contained in the genetic material of the original cell. On a larger scale, the progeny of a vertebrate animal share a striking resemblance to their parents, also the result of their inheritance of parental genes. A capacity to change over time by gradual evolution. Organisms change their inherited life strategies in very small steps to survive in new circumstances. The result of eons of evolution is an enormous diversity of life form, superficially very different, figure 1 से लेकर के 2, but fundamentally related through their shared ancestry. This fundamental unit of a living organism is reflected at the molecular level in the similarity of gene sequence and protein structure. Despite the common properties and the fundamental unity of life they reveal, it is difficult to make generalization about living organism. Earth has an enormous diversity of organisms, the range of habitats from hot spring to Arctic tundra, from animal intestine to college dormitories is matched by a correspondingly wide range of specific biochemical adaptation achieved within a common chemical framework. For the sake of clarity in this book, we sometimes risk certain generalization which, though not perfect, remains useful. We also frequently point out the exception to these generalization which can prove illuminating. Biochemistry describes in molecular terms the structure, mechanism, and chemical processes shared by all organisms and provide organizing principles that underlie life in all its diverse forms. Principle we refer to collectively as the molecular logic of life. Although biochemistry provides important insight and practical application in medicine, agriculture, nutrition, industry, its ultimate concern is with the wonder of life itself. In this introductory chapter, we give an overview of the cellular, chemical, physical, and genetic backgrounds to biochemistry and the overarching principle of evolution, how life emerged and evolved into the diversity of organisms we see today. As you read through the book, you may find it helpful to refer back to this chapter at interval to refresh your memory of this background material. So, number 1.1 cellular foundation. The unity and diversity of organism become apparent even at the cellular level. The smallest organism consist of single cells and are microscopic. Larger multicellular organisms contain many different type of cell which vary in size, shape, and specialized function. Despite these obvious differences, all cells of the simplest and most complex organism share certain fundamental properties which can be seen at the biochemical level. Cells are the structural and functional unit of all living organisms. Cell of all kinds share certain structural feature. Figure 1 से लेकर के 3. The plasma membrane defines the periphery of the cell, separating its content from the surroundings. It is composed of a lipid and a protein molecule that form a thin top pliable hydrophobic barrier around the cell. The membrane is a barrier to the free passage of inorganic ion and most other charged or polar compounds. Transport protein in the plasma membrane allow the passage of a certain ion and molecules. Receptor protein transmits signal into the cell. Membrane enzyme participate in some reaction pathway. Because the individual lipids and protein of the plasma membrane are not covalently linked, the entire structure is remarkably flexible, allowing changes in the shape and size of the cell. As a cell grows, newly made lipid and protein molecule are inserted into its plasma membrane. Cell division produces two cells, each with its own membrane. This growth and cell division मतलब fission occurs without loss of membrane integrity. The internal volume enclosed by the plasma membrane, जिसको हम साइटोप्लाज्म कहते हैं, फिगर 1 से लेकर के 3, is composed of an aqueous solution, जिसको हम साइटोजॉल बोलते हैं, and a variety of suspended particles with specific functions. These particulate components membranous organelles such as mitochondria and chloroplast, supramolecular structures such as ribosomes and proteosomes, the site of protein synthesis and degradation. ठीक है, the site of protein synthesis. Okay. These particulate components these particulates component sediment when cytoplasm is centrifuged at 1,50,000 grams. Okay, gram nahi, okay. G is the gravitational force. Okay, G का मतलब ग्राम नहीं, sorry for that. G is the gravitational force of earth. What remains as the supernatant fluid is the cytosol, a हाईली के बाद कुछ नहीं, इधर आओ, highly concentrated solution containing enzyme and the RNA molecules that encode them. The components मतलब amino acid and nucleotide from which these macromolecules are assembled, hundred of small organic molecules called metabolites intermediates in biosynthesis and degradative pathways. Coenzymes, compound essential to many enzyme catalyzed reaction and inorganic ions. All cells have, for at least some part of their life, either a nucleus or a nucleus in which the genome, the complete set of a gene composed of a DNA, is replicated and stored. With its associated protein, the nucleus in bacteria and archaea is not separated from the cytoplasm by a membrane. The nucleus in the eukaryotes is enclosed within a double membrane, the nuclear envelope. Cells with nuclear envelope make up the large domain eukarya. Eu means true, karyon means nucleus. Microorganisms without nuclear membrane formerly grouped together as prokaryotes, Greek word pro means before, are now recognized as comprising two very distinct groups, the domain bacteria, archaea described below. Cellular dimensions are limited by diffusion. Most cells are microscopic, invisible to the unaided eye. Animal and plant cells are typical 5 से लेकर के 100 micrometer in diameter, and many unicellular microorganisms are only 1 से लेकर के 2 micrometer long. See the inside back cover for information on the units and their abbreviation. What limits the dimension of a cell? The lower limit is probably set by the minimum number of each type of biomolecules required by the cell. The smallest cell, certain bacteria known as mycoplasm, are 300 nanometer in diameter and have a volume of about 10 to the power minus 14 ml. A single bacterial ribosome is about 20 nanometer in its longest dimension, so a few ribosomes take up a substantial fraction of the volume in a mycoplasmal cell. The upper limit of cell size is probably set by the rate of diffusion of a solute molecule in aqueous systems. For example, a bacterial cell that depends on oxygen consuming reaction for energy extraction must obtain molecular oxygen by diffusion from the surrounding medium through its plasma membrane. The cell is so small and the ratio of its surface area to its volume is so large that every part of its cytoplasm is easily reached by oxygen diffusing into the cell. With increasing cell size, however, surface to volume ratio decreases until metabolism consumes oxygen faster than diffusion can supply it. Metabolism that requires oxygen thus becomes impossible as cell size increases beyond a certain point, placing a theoretical upper limit on the size of the cell. Oxygen is only one of many low molecular weight species that must diffuse from outside of the cell to various regions of its interior, and the same surface to volume argument applies to each of them as well. There are three distinct domains of life. There are three distinct domains of life. All living organisms fall into one of the three large groups जिसको हम डोमेन कहते हैं, that defines three branches of the evolutionary trees of life originating from a common progenitor. फिगर 1 से लेकर के 4 जो कि ये है। ओके, ये फिगर को बाद में देखेंगे, पहले रीडिंग मार लेते हैं। Two large group of single-celled microorganisms can be distinguished on genetic and biochemical grounds: bacteria and archaea. Bacteria inhabit soils, surface water, and the tissue of other living or decaying organisms. Many of the archaea recognized as a distinct domain by Carl Uch in 1980 inhabit extreme environment, that is salted lake, hot spring, highly acidic bogs, and the ocean depth. The available evidence suggests that archaea and bacteria diverged early in evolution. All eukaryotic organisms which make up the third domain, that is eukarya, evolved from the same branch that gave rise to the archaea. Eukaryotes are therefore more closely related to archaea than to bacteria. With the domain of archaea and bacteria are subgroup distinguished by their habitats. In aerobic habitats with a plentiful supply of oxygen, some resident organisms derive energy from the transfer of electron from the fuel molecules to oxygen within the cell. Other environment are anaerobic, virtually devoid of oxygen, and microorganisms adapted to these environment obtain energy by transferring electron to nitrate, that is forming N2, sulfate forming hydrogen sulfide, or CO2 forming CH4. Many organisms that have evolved in anaerobic environment are obligate anaerobes. They die when exposed to oxygen. Others are facultative anaerobes, able to live with or without oxygens. Organisms differ widely in their source of energy and biosynthesis synthetic precursor. We can classify organisms according to how they obtained the energy and carbon they need for synthesizing cellular material as summarized in figure 1 से लेकर के 5. There are two broad category based on energy source. Phototrophs, ग्रीक वर्ड ट्रोप का मतलब होता है नॉरिशमेंट, ट्रोफी, trap and use sunlight, and chemotrophs derive their energy from oxidation of a chemical fuel. Some chemotrophs oxidize inorganic fuel HS to S0 या नॉट, इसे पता नहीं क्या बोल रहे हैं, इसे एलिमेंटल सल्फर भी बोलते हैं। S0 to और ये क्या है, यहाँ पर सेंटेंस गायब है, पता नहीं। फॉर एग्जाम्पल यहाँ पर मिसिंग है। Phototrophs and chemotrophs may be further divided into those that can synthesize all their biomolecules directly from CO2, that is autotrophs, and those that require some preformed organic nutrients made by other organism, that is heterotrophs. We can describe an organism mode of nutrition by combining these terms. For example, cyanobacteria are photoautotrophs. Humans are chemoheterotrophs. Even finer distinction can be made, and many organisms can obtain energy and carbon from more than one source under different environmental or developmental conditions. Bacteria and archaeal are cells share common feature but differ in important ways. The best studied bacterium, Escherichia coli, is usually harmless inhabitant of the human intestinal tract. The Escherichia coli cell is an ovoid about 2 micrometer long and a little less than 1 micrometer in diameter, but other bacteria may be spherical or rod shaped. It has a protective outer membrane and the inner plasma membrane that encloses the cytoplasm and the nucleids. Between the inner and outer membrane is a thin but strong layer of high molecular weight polymer, जिसे हम पेप्टिडोग्लाइकन कहते हैं, that gives the cell its shape and rigidity. The plasma membrane and the layer outside it constitute the cell envelope. It constitute the cell envelope. The plasma membrane of a bacteria consists of a thin bilayer of lipid molecule penetrated by proteins. Archaeal plasma membrane have a similar architecture, but the lipids can be strikingly different from those of bacteria. Bacteria and archaea have group specific specialization of their cell envelope. Some bacteria called gram positive because they are colored by gram stain introduced by Hans Peter Gram in 1882 have a thick layer of peptidoglycan outside their plasma membrane but lack an outer membrane. Gram negative bacteria have an outer membrane composed of a thin lipid bilayer into which are inserted complex lipopolysaccharides and proteins called porins that provide transmembrane channel for low molecular weight compound and ions to diffuse across this outer membrane. The structure outside the plasma membrane of archaea differs from the organism to organism, but they too have a layer of peptidoglycans or protein that confirms rigidity on their cell envelope. सेल एनवलप खत्म हो गया. Then the cytoplasm of Escherichia coli contain about 15,000 ribosomes, various number 10 to 1,000 of copies of each of 1,000 or so different enzyme, perhaps 1,000 organic compounds of molecular weight less than 1,000 metabolites and cofactor, and a variety of inorganic ions. The nucleids contain a single circular molecules of DNA, and the cytoplasm, like that of most bacteria, contains one or more smaller circular segment of DNA called as plasmids. In nature, some plasmid confers resistance to toxin and antibiotics in the environment. In the laboratory, these DNA segments are especially amenable to experimental manipulation and are powerful tools for genetic engineering. See chapter number 9. Other specific of bacteria as well as archaea contains a similar collection of biomolecules, but each species has physical and metabolic specialization related to its environmental niche and nutritional sources. Cyanobacteria, for example, have internal membranes specialized to trap energy from light. See figure 1967. Many archaea live in extreme environment and have biochemicals biochemicals adaptation to survive in extreme of temperature, pressure और salt concentration. ओके यहाँ पर थोड़ा इसको भी कवर लेते हैं. तो ऑल ऑर्गेनिज्म एनर्जी सोर्स क्या हो जाएगा? केमिकल एंड लाइट. केमिकल में केमोट्रोप्स, उसके बाद कार्बन सोर्सेस दो प्रकार से, कार्बन डाइऑक्साइड ऑर्गेनिक कंपाउंड से आता है. कार्बन डाइऑक्साइड से केमो ऑटोट्रोप्स होता है. देन इसमें केमोटोट्रॉप्स में हाइड्रोजन, सल्फर, आयरन, नाइट्रोजन, कार्बन मोनोक्साइड, ऑक्सीडाइजिंग बैक्टीरिया. देन ऑर्गेनिक कंपाउंड में केमो हेटेरोट्रॉप्स, फाइनल इलेक्ट्रॉन एक्सेप्टर, उसके बाद ऑक्सीजन, ऑल एनिमल, मोस्ट फंजाई, प्रोटिस्ट, बैक्टीरिया. देन फाइनल इलेक्ट्रॉन एक्सेप्टर में नॉट ऑक्सीजन होगा, ऑर्गेनिक कंपाउंड, देन इनऑर्गेनिक कंपाउंड. इनमें से फर्मेंटेटिव बैक्टीरिया सच एज लैक्टोकोकस लैक्टिस एंड उसके बाद कोई मेंशन नहीं. इनऑर्गेनिक कंपाउंड में स्यूडोमोनास, डेंट्रीफिकेंस फॉर एग्जांपल. यहाँ तक ये कंप्लीट हो गया. लाइट में फोटोट्रॉप्स, उसका कार्बन सोर्स कार्बन डाइऑक्साइड एंड ऑर्गेनिक कंपाउंड. कार्बन डाइऑक्साइड से फोटोट्रोप्स यूज वाटर टू रिड्यूस कार्बन डाइऑक्साइड. क्या ये करेक्ट है? क्वेश्चन मार्क है. अगर ये एस आएगा तो ऑक्सीजेनिक फोटोसिंथेसिस होगा, प्लांट अल्जे एंड सियानोबैक्टीरिया। अगर ये नो आएगा तो अनऑक्सीजेनिक फोटोसिंथेसिस बैक्टीरिया, जिसमें ग्रीन एंड पर्पल बैक्टीरिया आता है। देन ऑर्गेनिक कंपाउंड्स में फोटो हेटेरोट्रोप्स आएगा, ग्रीन नॉन सल्फर बैक्टीरिया, पर्पल नॉन सल्फर बैक्टीरिया। ये बस टेबल खत्म हो गया। Difference in ribosomal structure gave the first hint that bacteria and archaea constituted separate domain. Most bacteria, including Escherichia coli, exist as individual cell but often associated in biofilms or mats in which large number of cells adhere to each other and to some solid substrate beneath or at an aqueous surface. Cell of some bacterial species, the Myxobacteria, for example, shows simple social behavior, forming many celled aggregate in response to signals between neighboring cell. Eukaryotic cell have a variety of membranous organelle which can be isolated for study. टिपिकल यूकैरियोटिक सेल फिगर 1 से लेकर के 7 आर मच लार्जर दैन बैक्टीरिया, कॉमनली 5 से लेकर के 100 माइक्रोमीटर इन डायमीटर with cell with cell volume 1,000 to million times larger than those of a bacteria. The distinguishing characteristic of eukaryotes are the nucleus and a variety of membrane enclosed organelle with specific function. These organelles include mitochondria, the site of most of energy extracting reaction to the cell of the cell, the endoplasmic reticulum and Golgi complex, which play central role in the synthesis and processing of lipid and the membrane protein. Peroxisome, in which very long chain fatty acid are oxidized. Lysosome, filled with digestive enzyme to degrade unneeded cellular debris. In addition to these, plant cells also contain vacuoles which store large quantities of organic acid, and chloroplast, in which sunlight drives the synthesis of ATP in the process of photosynthesis. फिगर 1 से लेकर के 7 also present in the cytoplasm of many cells are granules or droplets containing stored nutrients such as starch and fat. यहाँ पर ये सारा का सारा डायग्राम है। बहुत बड़ा चैप्टर है क्या? काफी बड़ा है, बाप रे। वाओ, प्रॉब्लम्स दिया गया है, काफी बड़ा चैप्टर है। स्ट्रक्चर एंड कैटलिस्ट, ओके ठीक है। ओके, in a major advance in chemistry, Albert Claude, Christian D. Dube, and George Pallade developed a method for separating organelles from the cytosol and from each other, an essential step in investigating their structure and function. In a typical cell fractionation, figure 1 से लेकर के 8, cell or tissue in solution are gently disrupted by physical shear. This treatment ruptures the plasma membrane but leaves most of the organelle intact. The homogenate is then centrifuged. Organelles such as nuclei, mitochondria, lysosome differ in size and therefore sediment at different rates. These methods were used to establish, for example, that lysosome contain degradative enzyme, mitochondria contain oxidative enzyme, chloroplast contain photosynthetic pigments. The isolation of an organelle enriched in a certain enzyme is often the first step in the purification of that enzyme. The cytoplasm is organized by the cytoskeleton and is highly dynamic. Fluorescence microscopy reveals several types of protein filament crisscrossing the eukaryotic cell, forming an interlocking three-dimensional meshwork, the cytoskeleton. There are three general type of cytoplasmic filament: actin filament, microtubules and intermediate filament. Figure 1 से लेकर के 9, differing in width from about 6 to 22 nanometer, composition and specific function. All types provided structure and organization to the cytoplasm and shape to the cell. Actin filament and microtubules also help to produce the motion of organelles or of the whole cell. Each type of a cytoskeletal component is composed of a simple protein subunit that associated non-covalently to form filaments of a uniform thickness. These filaments are not permanent structure. They undergo constant disassembly into their protein subunit and the reassembly into filaments. Their location in the cells are not rigidly fixed, but many change dramatically with mitosis, cytokinesis, amo, amoeboid motion, or change in cell shape. The assembly, disassembly, and the location of all type of filaments are regulated by other protein which serve to link or bundle the filaments or to move cytoplasmic organelles along the filaments. That is bacteria containing actin like protein that serve similar role in those cells. The picture that emerges from this brief survey of eukaryotic cell structure is of a cell with a meshwork of structural fiber and a complex system of membrane enclosed compartment. The filaments disassemble and then reassemble elsewhere. Membranous vesicles board from one organelle and fuse with another. Organelles move through the cytoplasm along protein filaments. Their motion powered by energy dependent motor proteins. The endomembrane system segregates specific metabolic processes and provides surface on which certain enzyme catalyzed reaction occurs. Exocytosis and endocytosis mechanism of transport out of and into cell respectively that involve membrane fusion and fission provide paths between the cytoplasm and the surrounding medium, allowing for secretion of substance produced in the cell and uptake of extracellular materials. ये काफ़ी बड़ा चैप्टर है. कम से कम 10 पेज पढ़ना पड़ेगा तभी हो पाएगा. Although complex, this organization of the cytoplasm is far from random. The motion and positioning of organelles and cytoskeleton element are under tight regulation, and at certain stages in its life, a eukaryotic cell undergoes dramatic, finely orchestrated uh, reorganization, such as the event of mitosis. The interaction the interaction The interaction between the cytoskeleton and organ are non-covalent, reversible, and subject to regulation in response to various intracellular and extracellular signals. Cell build supramolecular structure. Macromolecules and their monomeric subunit differ greatly in size. An alanine molecule is less than 0.5 nanometer long. A molecule of hemoglobin, the oxygen carrying protein of erythrocyte red blood cell, consists of nearly 600 amino acid subunit in four long chain folded into globular shape and associated in a structure 5.5 nanometer in diameter. In turns, proteins are much smaller than ribosomes, about 20 nanometer in diameter, which are in turn much smaller than organelles such as mitochondria, typically 100 nanometer in diameter. It is a long jump from simple biomolecules to cellular structure that can be seen with the light microscope. Figure 1 से लेकर के 11 illustrate the structural hierarchy in cellular organization. The monomeric subunit of a protein, nucleic acid, and a polysaccharide are joined by covalent bond in supramolecular complexes. However, macromolecules are held together by non-covalent interaction much weaker individually than covalent bonds. Among these non-covalent interactions are hydrogen bonds between the polar groups, ionic interaction between charged group, hydrophobic interaction among non-polar groups in aqueous solution, and van der Waal interaction, that is London forces, all of which have energies much smaller than those of the covalent bond. These non-covalent interactions are described in chapter number 2. The large number of weak interactions between the macromolecules in supramolecular complexes stabilize these assemblies, producing their unique structure. In vitro studies may overlook important interaction among the molecule. In vitro studies may overlook important interaction among the molecules. One approach to understanding a biological process is to study purified molecules in vitro, that is, in glass, in the test tube, without interference from other molecules present in the intact cell, that is, in vivo in the living. In vivo in the living. इन vivo, vivo मतलब मोबाइल, मोबाइल को मैन यूज करता है, तो इन vivo. Although this approach has been remarkably revealing, we must keep in mind that the inside of a cell is quite different from the inside of the test tube. The interfering component eliminated by purification may be critical to the biological function or regulation of the molecule purified. For example, in vitro studies of a pure enzyme are commonly done at a very low enzyme concentration in thoroughly stirred aqueous solution. In the cell, an enzyme is dissolved or suspended in the gel-like cytosol with thousands of other proteins, some of which bind to that enzyme and influence its activity. Some enzymes are components of a multi-enzyme complex in which the reactant are channeled from one enzyme to another, never entering the bulk solvent. When all of known macromolecules in a cells are represented at their known dimension and concentration, it is clear that the cytosol is very crowded and that diffusion of macromolecules within the cytosol must be slowed by collisions with other large structure. In short, a given molecule may behave quite differently in the cell and in vitro. A central challenge of biochemistry is to understand the influence of cellular organization and macromolecular association on the function of individual enzyme and other biomolecules. To understand function in vivo as well as in vitro. Summary 1.1. Cellular foundation. All cells are bounded by a plasma membrane, have a cytosol containing metabolites, coenzyme, inorganic ions, enzyme, and have a set of genes contained within a nucleates. बैक्टीरिया आर्किया न्यूक्लियस जो कि यूकैरियोट में होता है. All organisms require a source of energy to perform cellular work. Phototrophs obtain energy from sunlight. Chemotrophs oxidize chemical fuels, passing electrons to good electron acceptor, inorganic compounds, organic compounds, and molecular oxygens. Bacteria and archaeal cell contain cytosol, a nucleoid, and plasmids all contained within a cell envelope. Eukaryotic cells have a nucleus and are multi-compartmented with certain processes. फिगर 1 से लेकर के 10. ओके बाप रे बाप रे. सम कंपोनेंट ऑफ लिपिड. ठीक है. फिगर 1 से लेकर के 10. द ऑर्गेनिक कंपाउंड फ्रॉम व्हिच मोस्ट सेलुलर material are constructed. The ABC of biochemistry. Shown here are A for 6 of the 20 amino acid from which all protein are built. The side chain are shaded light red. B. The 5 nitrogenous bases, 2 5 carbon sugar and phosphate ion from which all nucleic acid are built. C 5 component of the membrane lipid. D मतलब D glucose, a simple sugar from which most carbohydrate are divided. Note that phosphate is a component of both nucleic acid and membrane lipid. Segregated in specific organelles. Organelles can be separated and studied in isolation. Cytoskeletal protein assemble into long filaments that gives cell shape and rigidity and serve as rail along which cellular organelles moves throughout the cell. supramolecular complex held together by non-covalent interaction are part of hierarchy of structure, some visible with light microscope. When individual molecules are removed from these complexes to be studied in vitro, interaction important in the living cell may be lost. Now coming to the chemical foundation. Biochemistry aims to explain biological form and function in chemical terms. By the late 18th century, chemistry had concluded that the composition of living matter is strikingly different from that of the inanimate world. Anthony Laurent Lavoisier 1743 से लेकर के 1794 noted the relative chemical simplicity of the mineral world and the contrast contrasted it with the complexity of the plant and animal world. The latter, he knew, were composed of a compound rich in the element of carbon, oxygen, nitrogen, and phosphorus. During the first half of the 20th century, uh, parallel biochemical investigation of a glucose breakdown in the yeast and in animal muscle cells revealed remarkable chemical similarity in these two apparently very different cell types. The breakdown of glucose in the yeast and muscle cell involved the same 10 chemical intermediates and the same 10 enzyme. Subsequent studies of many other biochemical processes in many different organisms have confirmed the generality of this observation, neatly summarized in 1954 by Jacques Monod. What is true of Escherichia coli is true of the elephant. The current understanding that all organisms share a common evolutionary origin is based in part on this observed universality of chemical intermediates and transformation, often termed as biochemical unity. Fewer than 30 of more than 90 natural occurring chemical elements are essential to organism. Most of the elements in living matters have relatively low atomic number. Only 3 have atomic number above that of selenium, जो कि 34. The four most abundant element in living organism in term of a percentage of a total number of atom are hydrogen, oxygen, nitrogen, carbon, which together make up more than 99 percentage of the mass of the most cell. They are the lightest element capable of efficiently forming 1, 2, 3, and 4 bonds respectively. In general, the lightest element form the strongest bond. The trace element represent a minuscule fraction of the weight of the human body, but all are essential to life, usually because they are essential to the function of a specific protein, including many enzymes. The oxygen transporting capacity of the hemoglobin molecule, for example, is absolutely dependent on four iron ions that make up only 0.3 percentage of its mass. Biomolecules are compounds of carbon with a variety of functional groups. The chemistry of a living organism is organized around carbon, which account for more than half the dry weight of the cell. Carbon can form single bond with hydrogen atom, both single and double bond with oxygen and nitrogen atom. of greatest significance in biology is the ability of a carbon atom to form very, very stable, very stable, very stable single bond with up to four other carbon atom. Two carbon atom also can share two or three electron pairs, thus forming double or triple bond. The four single bond that can formed by a carbon atom project from the nucleus to four apices, four pieces of a tetrahedron with an angle of about 109.5 degree between any two bonds and an average bond length of 0.154 nanometer. There is a free rotation around each single bond unless very large or highly charged groups are attached to both carbon atoms, in which case rotation may be restricted. A double bond is shorter, about 0.134 nanometer, and rigid and allows only limited rotation about its axis. Covalently linked carbon atom in biomolecules can form linear chains, branched chain, and cyclic structure. It seems likely that the bonding versatility of carbon with itself and with other element was a major factor in the selection of a carbon compound for the molecular machinery of cell during the origin and evolution of living organisms. No other chemicals element can form molecule of such widely different size, shape, and composition. Most biomolecule can be regarded as the derivative of a hydrocarbon, with hydrogen atom replaced by a variety of a functional group that conform specific chemical properties on the molecules, forming various families of organic compounds. Typical of these are alcohol, which have one or more hydroxyl group, amines with amino group, aldehyde ketones with carbonyl groups, and carboxylic acid with carboxyl groups. Many biomolecules are polyfunctional, containing two or more type of functional groups. फंक्शनल ग्रुप्स कहाँ पर ये खत्म हुआ? फंक्शनल ग्रुप यहाँ पर नहीं, फिगर सी। ओके, ये सब एक्सप्लेन है शायद। Figure, each with its own chemical characteristic and reaction. The chemical personality of a compound is determined by the chemistry of its functional group and their disposition in three-dimensional space. तो आज हम इसको यहाँ पर एंड करते हैं। आज का डेट कितना है? 4 फरवरी 26. Thanks for watching.